Hello, welcome to Biology, the scientific study of life. Cloning and expression eukaryotic genes. As an alternative to screening a DNA library of a particular nucleotide sequence, the clones can sometimes be screened for a desired gene based on detection of its encoded protein. For example, if the protein is an enzyme, its activity can be measured. Alternatively, the protein can be detected using antibiotics that bind specially to it. And once a particular gene has been cloned in host cells, its protein product can be produced in larger amounts for research purposes or develop practical applications. Bacterial expression systems. Getting a cloned eukaryotic gene to function in bacterial host cells can be difficult because certain aspects of gene expression are different in eukaryotic and prokaryotism. To overcome differences in parameters and over DNA control sequences, scientists usually employs an expression vector, a cloning vector that contains a highly active prokaryotic parameters just upstream of a restriction site where the eukaryotic gene can be inserted in the correct reading frame. The bacterial host cell will recognize the parameter and proceed to express the foreign gene now linked to that parameter. Such expression vectors allow the synthesis of many eukaryotic proteins in bacterial cells. Another problem with expression cloned eukaryotic genes in bacteria is the presence of non-coding regions, introns, which means in most eukaryotic genes, introns can make a eukaryotic gene very long and they prevent correct expression of the gene by bacterial cells which do not have RNA splicing machinery. This problem can be surmounted by using a cDNA form of the gene, which includes only the exons. Bacteria can express a eukaryotic cDNA gene if the vector contains a bacterial parameter and any other control elements necessary for the gene's transcription and translation. Eukaryotic cloning and expression systems. Molecular biologists can avoid eukaryotic prokaryotic incompatibility by using eukaryotic cells such as yeasts rather than bacteria as hosts for cloning and or expressing eukaryotic genes of interest. Yeast cells, single-celled fungi offer two advantages. They are as easy to grow as bacteria and they have plasmids, a rarity among eukaryotes. Scientists have even constructed recombinant plasmids that combine yeast and bacterial DNA and can replicate in either type of cell. Another useful tool for cloning eukaryotic genes are yeast artificial chromosomes, short-term YACS, which combine the essential of a eukaryotic chromosome and origin for DNA replication, a centromere and two telomeres with foreign DNA. These chromosome-like vectors behave normally in mitosis, cloning the foreign DNA as the yeast cell divides. Because YAC can carry a much longer DNA segment than can a plasmid vector, a cloned fragment is more likely to contain an entire gene rather than just a protein of it, rather than just a portion of it. Another reason to use eukaryotic host cells for expressing a cloned eukaryotic gene is that many eukaryotic proteins will not function unless they are modified after translation, for example, by the addition of carbohydrate or lipid groups. Bacterial cells cannot carry out these modifications, and if gene product requiring such processing is from a mammal, even yeast cells will not be able to modify the protein correctly. The use of host cells from an animal cell culture may therefore be necessary. Scientists have developed a variety of methods of introducing recombinant DNA into eukaryotic cells in electroporation. Bon. A brief electrical bon. A brief practical or electrical pulse applied to a solution containing cells creates temporary holes in their plasma membrane through which DNA can enter. This technique is now commonly used for bacteria as well. Alternatively, scientists can inject DNA directly into single eukaryotic cells using microscopically thin needles. Notice that thin needles. This is the research. To get DNA into plant cells, the soil bacterium, agrobacterium, can be used as well later if the introduced DNA is incorporated into a cell's genome by genetic recombination, then it may be expressed by the cell. Amplifying DNA in vitro, the polymers chain reaction PCR DNA cloning in cells remains the best method of preparing large quantities of the particular gene or other DNA sequence. However, when the source of DNA is scanty or impure of the polymers chain reaction or PCR is quicker and more selective. In this technique, any specific target segment within one or many DNA molecules can be quickly amplified, copied, which means many times, in a test tube with automation. PRC or PCR can make a billion of copies of target segment of DNA in a few hours. Significantly faster than the days it would take to obtain the same number of copies by screening a DNA library for a clone with the desired gene and letting it replicate within host cells. In the PCR procedure, a three-step cycle, notice that this step or third one, brings about chain reaction that produces an exponentially can produce three cycle brings about a chain reaction that produces an exponentially growing population of identical DNA molecules. During each cycle, the reaction mixture is heated to denature, which means separate the DNA strands, and then cooled to allow annealing hydrogen bonding of short single-strand DNA primers complementary to sequence on opposite strand like these. Notice that. At each each end of the target sequence. Finally, a heat-stable DNA polymers extends the primers in the 5 to 3 direction. If a standard DNA polymers were used, the protein would be denatured along with the DNA during the first heating step and would have to be replaced after each cycle. The key to automating PCR was the discovery of an unusual heat-stable DNA polymers, first isolated from prokaryotes living in hot springs, that would withstand the heat and at the start of each cycle. Just as impressive as the speed of PCR is its specificity. Only minute amounts of DNA need be present in the starting material, and this DNA can be in a partially degraded state. The key to this high specificity is the primers, which hydrogen bond only to sequences at opposite ends of the target segment. By the end of the third cycle, 1/4 of the molecules are identical to the target segment, with both strands at the appropriate length. With each successive cycle, the number of target segment molecules of the correct length doubles, soon greatly outnumbering all other DNA molecules in the reaction. Despite its speed and specificity, PCR amplification cannot substitute for gene cloning in cells when large amounts of a gene are desired. Occasional errors during PCR replication impose limits on the number of good copies that can be made by this method. Increasingly, however, PCR is used to make enough of a specific DNA fragment to clone it merely by inserting into a vector. Devised in 1985, PCR has had a major impact on biological research and biotechnology. PCR has been used to amplify DNA from a wide variety of sources. Fragments of ancient DNA from 40,000 years old frozen woolly mammoth, DNA from fingerprints or from tiny amounts of blood, tissue, or semen found at crime scenes, DNA from single embryonic cells for rapid prenatal diagnosis of genetic disorders, a DNA of viral gene from cells infected with viruses that are difficult to detect, such as HIV. We will return to application of PCR later in the book chapter. Back to this research method, the polymers chain reaction PCR application. Notice that with PCR and specific segment, the target sequence within a DNA sample can be copied many times, amplified completely in vitro. Technique: the starting material for PCR, a double-stranded DNA containing the target nucleotide sequence to be copied ahead. Resistant DNA polymers, all 4 nucleotides, and too short. Notice that single-stranded DNA molecules that serve as primers is complementary one strand to one end of the target. Notice that cycle 1 molecules, 2 molecules, and 3 in white boxes match target sequence. The result is during each PCR cycle, the target DNA sequence is doubled by the end of the 3 cycle 1/4. The molecules correspond exactly to the target sequence with sequence or both strands of the correct length. Notice also, see the white boxes above. After 20 or so cycles, the target sequence molecules outnumber all others by a billion-fold or more. Notice that. Okay, thanks for listening.